This is lecture US-2, second lecture of the urinary system. In this lecture, we're going to start by talking about filtration, the structure of the glomerulus, what pressures are involved, and how we regulate that using the process of tubulo-glomerular feedback. In part B, we'll be looking at reabsorption in the nephron. We'll talk about how much is reabsorbed, where it's reabsorbed, some mechanisms in the proximal tubule, and then some of what goes on in the loop of Henle, distal tubule, and collecting duct. Finally, in part C, we'll talk about hormonal control of that reabsorption. Specifically, we'll talk about how aldosterone, vasopressin, and how those two are involved in regulating reabsorption, especially of water, and then the functioning of the renin-angiotensin-aldosterone system for regulating blood pressure in the body through water reabsorption and sodium reabsorption in the kidneys. Okay, let's talk filtration in the kidneys. Now, when we talk about filtration, one of the figures we're going to keep coming back to is this idea of glomerular filtration rate. Which we often just abbreviate GFR. We're going to be talking about things that affect the glomerular filtration rate, the rate at which fluid is moved from the blood into the urinary tubule at the glomerulus. The kind of the book standard, the value that you will see as the norm, is often about 180 liters per day, which is an enormous amount. If you figure there are about 4 liters per gallon, that's going toward 40 to 50 gallons per day filtering out of the blood and into the urinary tubule. Given that you've got maybe 6 liters of blood, the fact that you're filtering 180 liters a day implies that you're filtering out your entire blood supply over and over again over the course of a day, which is true. Which is why, when we talk about those 4 functions of filtration, reabsorption, secretion, and excretion, while we filter out a lot of blood, we have to reabsorb almost all of it, or you would lose your entire blood supply. So, this is the value we're going to use as typical for a GFR. Now, a lot of what we talk about today is going to be how we adjust this, how we change how much we're filtering up or down, which is one of our kidneys' more important jobs, is keeping that rate constant even when other things are going to change it. One reason we wouldn't want to change that is figuring I only have so much blood. My total blood volume doesn't really change very much, which means that I don't usually need to change how often I filter it. It just needs to go through that filter at a constant rate. But as you'll see, this filtration rate is largely determined by the pressure in the glomerulus, and that changes with our blood pressure. So one of the big things we talk about is how we're going to adjust glomerular filtration rate to keep it constant. And we're going to use negative feedback and mechanisms for that. But we're going to start now by talking about the structure of the glomerulus, how it's physically arranged, and then how our body then uses that to change glomerular filtration rate, and how that adjusts filtration. So let's get to that. Okay, so let's start by talking about the structure of the glomerulus. So what I've drawn up here, which hopefully you can see, is a sketch of the glomerulus and Bowman's capsule. We have our afferent arteriole bringing blood to the glomerulus, which then goes into these glomerular capillaries. These are fenestrated capillaries. If you remember, fenestrated capillaries are the ones that have pores that allow materials up to the size of a small protein to move in or out of the glomerulus pretty freely. Then that comes out on the efferent arteriole. If you remember from our drawing of the folded nephron, our Bowman's capsule here then led into the proximal convoluted tubule, we'll label that here, which then led into the nephron loop, which then came up with the upper part of the nephron loop becoming the distal convoluted tubule right about here, passing between the afferent and efferent arterioles. We'll get back to that. But let's talk about this Bowman's capsule and these fenestrated glomerular capillaries first. So the capillaries first have, let's draw a kind of a cross section here. We've got, if we imagine here's our capillary, the capillary is made of these endothelial cells. Those form the wall of the capillary. And since this is a fenestrated capillary, we can put these little pores in here. So that's kind of layer one. So our blood is here inside this capillary with those fenestrations, which are going to allow materials to move out. Now, just outside of that, we have the basal lamina, this layer of protein filaments. So the basal lamina acts as another layer of filter. If you imagine your fenestrated capillary as sort of like a colander with little holes in it allowing water to move out when you're draining your pasta, the basal lamina would be sort of like a layer of coffee filter paper on top of on the outside of that colander. So now to get out, stuff has to pass through the holes in the colander and through the smaller holes in the filter paper. So this is now limiting what can get out to pretty small things. A few very small proteins might still be able to get through that basal lamina. So there's actually one more layer of filtration on top of this, and that is what we call podocytes. Podocytes are cells which have lots of little tiny extensions called pedicels that wrap around these capillaries and sort of intermesh with each other. So you could draw that looking sort of like, no, that one doesn't have any ink. Let's try a different one. So here I'm drawing, this is sort of one podocyte. Then we'll draw another podocyte here. You could see how they sort of interlock like that. Let me bring up a picture. So what you're seeing right now is an electron micrograph of a mouse glomerulus. The scale on the bottom shows you a 5-micron size bar. So you get an idea of that the big sort of cylindrical thing you're seeing is the capillary, and then the little things that are wrapped around it, those are the podocytes. You could see how the little pieces sort of intermesh with each other. Now, coming back to this, the spaces where those podocytes intermesh, so right here, that is a filtration slit, sometimes known as a slit pore. And in order to get out of the capillary, things have to now fit not just through the capillary fenestrations and the basal lamina, but also through that slit pore. And there's even another layer of filtering on top of that pore, sort of a membrane that sits on top of that. So if I were going to add that to our diagram down here, these interlocking pieces would look like this. So here's one going, for example, that way, and here's another one coming this way. Between them, this is the filtration slit. And there's actually a membrane in there covering those slits. So there's multiple layers of filtration here. In order to get out of the blood, we pass through the fenestrations, the basal lamina, and then between the pedicels of the podocytes through a filtration slit and through the membrane in that slit. So we've got multiple layers of paper, filter paper on top of all this, which limits the size of things that can get out. But by the time you get through all of this, really there should be virtually no protein and certainly no cells penetrating this. But anything smaller than that, so water, ions, small organic molecules like glucose or amino acids, all of those filter out freely. They can fit through all of those layers of filtration. So when our blood is leaving these capillaries, anything smaller than protein, and sometimes a few small proteins, can come out, and we get about 20% of it filtering out from the blood vessels into the Bowman's capsule. We know that those podocytes are important. There are people who are born with genetic problems with those podocytes, and their kidneys don't work properly at all. They filter out way too much. Both too much stuff and too big of stuff comes filtering out, so they run into some kidney problems. Now, there is another type of cell in here called the mesangial cells. I'm just going to sort of draw those in here. We used to think that mesangial cells could act to sort of squeeze these capillaries and change the amount of filtration that goes on. There is some controversy about whether that's the case or not. They can squeeze, whether it matters or not is the controversy. So one thing we do know is that these mesangial cells kind of clean these filters. As stuff filters out, some larger materials like proteins can get stuck on those filters and clog them. The mesangial cells seem to be cleaning off those filters, making sure that they stay unclogged. So those are some of the physical things, the filtering that happens at the glomerulus that changes how we filter and make sure we're only filtering stuff that should be coming out. No, as little protein as possible, certainly no cells, but anything smaller than that can filter. All right, now that we've talked about that, let's talk about the pressures involved. So I'm going to erase some of these extra things. Don't really need to have those on here anymore. I'll leave that because it's not hurting anything. Now let's talk about what forces might cause water to move into or out of this glomerulus. So when we talked in capillaries about how extracellular fluid is produced, we talked about two forces acting on water. Hydrostatic pressure was pushing water out of the capillary, and then osmotic pressure from the proteins in the plasma was pulling water in. And if you remember on that diagram, we had hydrostatic pressure was pushing water out. It varied from about 35 on the arterial side of the capillary to about 15 millimeters of mercury on the venous side. Osmotic pressure pulling water in stayed at about 25 over the whole length of the capillary. So that meant that at one end we had a balance toward filtration, and the other end we had a balance toward absorption. This capillary, in theory, could do the same kind of thing, but we don't want this capillary to be absorbing. We want this capillary to always be filtering, for water to be moving out of it and carrying things like salts and small organics with it. So, how are we going to make sure that this capillary is always filtering? Well, if those are the two forces, hydrostatic pressure pushing out and osmotic pressure pulling in, pause for a moment and think, which pressure do I want to be higher if I want this thing to always be moving material out? It should be hydrostatic pressure. We want that to be as high as possible. And here's where the fact that we have two arterioles is going to come in handy. So, just for a moment, imagine this situation. Okay, so imagine that we have a pipe. And that pipe is going to have two valves on it. Here's our pipe. There's our two valves. Fluid is going to be moving that direction in the pipe. So each of these valves I can tighten or loosen. If I tighten it, it makes the tube narrower. If I loosen it, it makes it wider. And here, I'm going to put a little nick in the wall of the pipe so that water can come out of it. Now, my question is this. So if I've got water flowing through here, first, some water will be coming out. We'll put a little spray of water here. Now, if I wanted to make that spray bigger, one thing I could do is to send more water through the pipe. The more water I send through here, the more it's going to be spraying out. That makes sense, because the more water I put in here, the higher the pressure will be, and that pressure is what's pushing this out. So my question is this: if I'm not going to change the amount of water I send into the pipe, how could I make this spray bigger or smaller? Let's start with bigger. What could I do with these valves to make the spray bigger? Well, it seems to me that if the water's going this way, if I tighten that valve, that's going to make it harder for water to get out of this section of pipe, which will increase the pressure. So if I tighten this valve, that should increase the pressure and make my spray bigger. Tightening that produces that. If I make this smaller, it increases the pressure here, which will cause me to push more water out. What would happen if I tightened this valve? Well, that makes it harder for water to get into this, which will mean the spray will get smaller. It'll lower the pressure in here. Now, if you think about how that's going to be related to our glomerulus, in my glomerulus, what I've got is a pipe. My glomerular capillaries are the pipe. They've got fenestrations allowing water to come out. And I've got two valves. An afferent arteriole and an efferent arteriole. So blood's coming this way, just like here, water was going that way. If I want to make the pressure in here higher, which of these two valves do I want to tighten? Okay, so if we want to raise the pressure in these glomerular capillaries, we need to constrict the thing that allows fluid to come out of them. That's going to make fluid back up in these and cause higher pressure. Since blood is going in this direction, if we want pressure to be high in these glomerular capillaries, we want to tighten up the efferent arteriole, which will cause this to back up. And that is in fact what happens. The body keeps the efferent arteriole under some level of tonic constriction. Tonic meaning constant, not letting go. So since this particular arteriole stays under this tonic constriction, that's going to raise the pressure in here. Now here I'm going to lie to you and say that the efferent arteriole just stays tonically constricted all the time and the body doesn't mess with it. That is not true. The body does adjust the constriction of the efferent arteriole, but it's going to do that relatively little. That's going to stay relatively constant. And the afferent arteriole is the one that our body's going to mess with more, dilating it or constricting it to adjust the pressure in here. So to keep it simple, we're going to pretend that this just stays as it is all the time. Just keep in mind, if you ever study this more, that the efferent arteriole does get adjusted to. Alright, so if our efferent arteriole stays constricted, that means the pressure in here is going to be higher, since blood coming in will tend to back up in here. The outward hydrostatic pressure, we'll label this hydrostatic, is on the scale of about 50 millimeters of mercury, compared to 35 at the highest in a regular capillary. So we've upped the pressure here to where there's a lot of pressure pushing water out. Now, just like in the regular capillaries, we just also have to account for the fact that there's protein in here, but not out here. The protein should not be filtering out of these capillaries, which means we still have osmotic pressure pulling water back in. And just like in a regular capillary, that amounts to about 25 millimeters of mercury of pressure pushing water back. So if I just look at those, 50 millimeters of mercury outward pressure, 25 millimeters of mercury inward pressure. Which one's greater? Well, obviously the outward. This would suggest that we're going to be pushing water out of this thing really fast. And we are. But there is actually, in this case, there's one more pressure we have to consider. In a regular capillary, we didn't have to think about it. We just imagined that as water left the capillary, it went off into the extracellular fluid, which we kind of imagined as a infinitely large space, where there's no limit to how much we can put in there, so it doesn't build up any pressure pushing back. That's more or less true for extracellular fluid under most circumstances. But here, the water coming out of this capillary is coming into this relatively small space, this little Bowman's capsule, which is not very big. Yes, there's a drain, but still, water's going to build up in here and push back a little bit. We call that fluid pressure. And that amounts to about 15 millimeters of mercury of pressure pushing back. It's kind of like a reverse hydrostatic pressure. This comes from the pressure in the blood pushing out. This comes from the pressure buildup in the Bowman's capsule pushing back. So now let's look at which way water is going to go. My total outward pressure is about 50 millimeters of mercury, but I have two inwards of 25 and 15 each. If I add those together, the total inward pressure is about 40. So an outward pressure of 50, inward pressure of 40, which one's greater? Outward pressure of 50. So I have an overall balance toward water moving out here, which produces filtration. Now I that's the main take-home message here. If you have a couple of spare neurons, consider this, because this will be useful later. If I've tightened this to make sure the pressure here is high, what does that do to the pressure downstream? Since blood is having a harder time getting through here, the hydrostatic pressure downstream of that efferent arteriole is going to be low, which means that just like these capillaries are now biased toward filtration, any capillaries that come after that are going to be biased toward absorption. That's going to come in handy. Keep that in mind. Anyway, so those are the pressures governing filtration here at the glomerulus, and we've adjusted this by tightening this up to make hydrostatic pressure high to make sure this is constantly filtering out. But now we get to the point of, what do I do to adjust this pressure? Because notice, this hydrostatic pressure is based on blood pressure. If my blood pressure overall in the whole body goes up, that means more pressure coming in, which means that's going to be higher. Which means that I'm going to be filtering too much. Likewise, if my body blood pressure drops, pressure coming in is lower, which means that's going to be lower, which means I may not be filtering enough. I want that pressure to stay just about the same all the time if I can manage it. And if I'm pretending that this doesn't get adjusted, how can I change this pressure? The easiest way would be to adjust the afferent arteriole. If I constrict the afferent arteriole, think about what that's going to do to the pressure in here. Do think about that for a second. Okay. If I tighten this and make it harder for blood to get into the glomerulus, that's going to lower the pressure in here and reduce my filtration. Likewise, if I dilate this, that's letting more blood into the glomerulus, which will raise this. So I can use the afferent arteriole to adjust filtration rate. Now, to do that, I need to know when to dilate it or when to constrict it. And if I want to keep my filtration rate constant, there's a couple ways I might want to do that. To answer that, we're going to now talk about a phenomenon which has the most beautiful name in all of physiology. The phenomenon is tubulo-glomerular feedback. I'll put it up here. You can see it. Now, tubulo-glomerular feedback, this feels good to say, uses a machine that we call, another good name, the juxtaglomerular apparatus. Isn't that beautiful? So let's put that up too. All right. Now, in just a moment here, I'm going to erase this and then sketch out a little bit about how that's going to work. So hold tight. All right, let's talk tubuloglomerular feedback. Now, you may want to pause and just copy this part down. That's our folded up nephron, same diagram as you did before. And then this right here is just a close-up of this area, because we're going to be talking about some stuff there in a little more detail. So if you want, pause and copy those things down. All right, now, first, let me start over here so we can talk about some of the names of the things in here. Now, when we talked about the unfolded versus folded nephron, we mentioned that there's an area right here where the loop of Henle is coming up and becoming the distal convoluted tubule that's important and has to talk to the afferent arteriole. So let's look at that on the close-up here. That area on the ascending part of the nephron loop as it turns into the DCT is called the macula densa. Macula densa, which I will sometimes just abbreviate MD. The macula densa, how exactly it works we're not going to get into, but I'm going to summarize it right now and just say that it acts like a flow detector that monitors how much stuff is flowing through the ascending loop of Henle. Now, here at the afferent arteriole, there's two things I want to talk about. One of them is the basic smooth muscle that's around that afferent arteriole. But I do want to specifically call out a little bit of that smooth muscle here. This stuff, this is a modified kind of smooth muscle that we're going to call juxtaglomerular juxtaglomerular cells. You may also see these called granular cells in some sources. The juxtaglomerular cells are modified smooth muscle that can secrete a protein called renin, or renin, depending on who you ask. R-E-N-I-N. So let me just sketch those in up here. So here's my juxtaglomerular cells, and here is my macula densa. We want those to be right next to each other because they're going to communicate with a paracrine messenger, which, if you remember, only travels over very, very short distances. So, these things together, the macula densa and the juxtaglomerular cells, together they are known as the juxtaglomerular apparatus, which hopefully I just put up here on the screen. The juxtaglomerular apparatus, or JGA, as it's sometimes called. So the juxtaglomerular apparatus does this job of tubuloglomerular feedback. And here's how it works. The macula densa detects flow. Actually, let me erase some of this stuff to give me a place to write on here. So hopefully you've got this thing right here. So let me get rid of this. Okay. So our macula densa detects flow in the ascending loop of Henle. So imagine that our glomerular filtration rate goes down, probably because our blood pressure goes down. So let's say body blood pressure decreases, which causes glomerular filtration rate to decrease. That's going to be how we start here. So my pressure overall goes down, which means the filtration rate gets smaller. So less fluid is filtering out, which means there will be less flow going by the macula densa. So this causes a flow decrease. The macula densa detects decreased flow. When the macula densa detects decreased flow going past here, it's going to secrete a chemical signal. In that case, prostaglandin secretion. So the macula densa will secrete prostaglandins, which will cause dilation of the afferent arteriole. So if I dilate the afferent arteriole, that means more blood will be entering here and it will increase glomerular filtration rate. So more blood into glomerulus, which causes increase in GFR, which will make more flow come by the macula densa. You may notice that that looks familiar. That's a negative feedback system. We'll draw it up as a negative feedback system in just a moment. So, now I do want to also mention one other thing. You're just going to need to kind of put a note by this in your notes and say, make sure to remember this and come back to it. In this case, when the macula densa detects decreased flow, that prostaglandin also causes the juxtaglomerular cells to secrete renin. So start making a list to yourself of things that make those cells secrete renin. Number one, decreased flow past the macula densa. So I don't have room to write that on here, but you should build this list. Things that make juxtaglomerular cells secrete renin. Number one, decreased flow past macula densa, which could come as a result of low blood pressure. Make a note of that too. All right, so that's one side of tubuloglomerular feedback. If we were going to write this in our negative feedback model that we've sometimes had, so remember we've got our sensor. Our sensor is the macula densa, and what it detects is flow in the loop of Henle. So in the macula densa, we've got a set point, and if flow, so if the flow is low, we are going to secrete prostaglandins, which dilate afferent arteriole, which causes increased filtration, which will increase the amount of flow through the loop. So if the flow is too low, we do something to increase it. That's part of our negative feedback system. Now that we should also have something about what if flow is high? What if we detect too much flow past the macula densa? So put this aside. Now we're going to draw in another sequence of events of what if the flow is too high? So if the macula densa detects increasing flow in the loop. For example, if blood pressure is too great and now we're filtering too much. So this suggests GFR is too high. In that case, the macula densa secretes a different paracrine messenger. In this case, as it turns out, it's ATP. This is not for energy use. This is actually going to work as a signal, as a paracrine messenger. That causes the smooth muscle of the afferent arteriole to constrict. That will reduce pressure in the glomerulus, which will reduce GFR, which will reduce flow at the macula densa. There's the other side of our negative feedback loop. So there it is in written form. If we were going to go back in and add it in our negative feedback system, it might look like this. So this was our macula densa. And before we had, if flow was low, we secreted prostaglandins, which cause dilation of the afferent, which increases GFR, which increases flow at the loop. So what if the flow is high? Well, that caused ATP secretion, which caused constriction of the afferent, which lowers GFR, which lowers flow at the loop. So we have a complete negative feedback system. Yay! That is how, as my blood pressure goes up and down, if for some reason my blood pressure in my body goes up, I don't want that to produce too much filtration. So blood pressure goes up, I start filtering more, that causes too much flow, which means I will constrict the afferent arteriole and decrease the filtration rate. If my blood pressure drops, flow is not enough, so I secrete prostaglandins, which dilates the afferent arteriole and increases glomerular filtration rate. It's a nice balancing system that makes sure my GFR stays more or less constant. It also is going to factor into something else. And that's where your list of things that cause renin secretion is going to be involved. Because we were saying, well, what's the renin for? We'll get to it. So before we get there, I want to point out another couple things that happen here. Those juxtaglomerular cells will secrete renin in the case where there's the macula densa says there's not enough flow and it puts prostaglandins onto them. But there's two other things that will cause renin secretion. If the afferent arteriole is under less stretch, if there's not as much stretch there, that will cause the juxtaglomerular cells to secrete renin. A third thing is that those juxtaglomerular cells have beta-1 receptors. So if the sympathetic nervous system is causing epinephrine release, that will act on the beta-1 receptors and cause renin secretion. So on your list of things that cause renin secretion, 1, prostaglandins from the macula densa as a result of not enough flow through the loop of Henle. Number 2, less stretch in the afferent arteriole, which would come as a result of low blood pressure. Number 3, sympathetic stimulation to this area through beta-1 receptors on the juxtaglomerular cells. All of those will produce renin secretion. Now, I'm not going to tell you just yet what renin does, but if you think about it, we have 2 things involving low blood pressure which cause its secretion, and a third thing involving sympathetic stimulation. What do you think renin is going to do? If the things that make it are mostly around low blood pressure, what do you think renin is going to cause? Think in a negative feedback way. All right, we'll get to that. Our next topic is going to be reabsorption.